back with your next class. In today's class, we are going to discuss the anatomy of roots. Okay. Now, regarding the anatomy of roots, I must tell you one thing, that all this while, the entire chapter that we have discussed, already you have learned everything regarding the plant body. What are the distinct layers present inside the plant body? What is the composition of those layers? All of that you have learned. So today's class is going to be just a revision. All that information that I have given you all this while, we'll be gathering them and we'll be learning about anatomy of roots. If you remember all those, prior informations, in that case, understanding today's class would become very, very easy for you. Okay, so let us start. So first, we'll start with dicot roots and then eventually we'll discuss the monocot root. So first, regarding dicot root. See, the dicot root begins with the outermost layer, that is epidermis. Now, in case of roots, we also can call it to be epiblema to differentiate it from the stem. Okay, for easy differentiation, we can also call it epiblema. All right. So, epidermis or epiblema in the dicot roots is going to be single layered parenchymatous and compactly arranged cells. Okay, and there also will be various outward projections that would be present in the epidermis, and they of course would be the root hairs. Root hairs would be numerous and they would be increasing the surface area so that the roots can efficiently absorb water and minerals. Alright. Now, internal to the epidermis remains the cortex. Now, as you already know, cortex is tri-layered, right? Hypodermis, general cortex and endodermis. As already I have told you repeatedly, the hypodermis is absent in roots, be it dicot or be it monocot. So in that case, after the epidermis, there directly will be general cortex present. Alright. General cortex would be made up of various, or it will be always multi-layered and consist of parenchyma cells. Okay, parenchyma cells which will have thin cellulosic cell wall. Why I am mentioning the cell wall here is because the water that would be absorbed by the root here would now readily be passing through the cell wall of the general cortex, of the cells, parenchyma cells present in the general cortex. So from the root here, the water would now readily pass through the general cortex because of this parenchyma cells. Okay. So now, apart from the parenchyma cells being present here, they will be loosely packed. Internal to that would remain the endodermis, right? The endodermis, on the other hand, is always single layered and it is going to be very, very distinct. Okay. And it consists of barrel-shaped cells. Barrel-shaped cells, I think all of you remember, drum-like cells. Okay. Barrel-shaped cells. Now, this barrel-shaped cells, they are specialized because they have a cell wall made up of suberin, which was discovered by Kasperi. And hence, we call this to be Kasperian strip. Okay. Now, here is a very important information. See, endodermal cell wall, because they have suberin deposition, they are completely impermeable to water. They would not allow water to pass through them. So water will readily pass through the general cortex, but as the water reaches the endodermis, it would not be able to pass through the cell wall of the endodermal cells. That's a very important information. Later on, you will be using this information in the transport chapter. There you will be able to learn how the transport of root or a transport of materials inside the plant body would be taking place in details. That time, this information is going to be very, very important. Okay. Now, that's why endodermis is always single layered, barrel shaped, compactly arranged cells consisting of Casparian strips, which makes it impermeable to water. That's it. Now, internal to the endodermis remains the pericycle. Now, pericycle can be single to multi-layered, but then it is very, very important. It has a very, very important role to play in case of dicot roots. See, in dicot roots, the pericycle will later on during secondary growth phase. Okay, when the secondary growth phase will be reached, the plant is mature enough. At that time, a few cells of the pericycle will convert meristematic and give rise to cambium. If you remember from our previous discussion that cambium is a thin layer of meristematic cells present in dicot stem in between xylem and phloem, but it is not present in the roots from beforehand. In case of dicot roots, this cambium will be rising from the pericycle, but later on during secondary growth phase. Understood that? So do remember, the pericycle will be single to multi-layered, made up of parenchyma cells. Later on it will help in giving rise to cambium, okay, in case of dicots. All right. Another important function of the pericycle is that the lateral roots will arise from the pericycle. Okay, if you remember in the morphology chapter, I told you the branches of the roots are endogenous, means they will arise from the pericycle. So pericycle here will also give rise to lateral roots. Okay. Now, internal to the pericycle will remain the vascular bundles. Now, as you already know, vascular bundles are going to be radial and exarch. Radial means the xylem and phloem are in alternation to one another. Xylem, next is phloem. Again a xylem and phloem, but in separate radii. And also they will be exarch, means metaxylem towards pit and protoxylem towards periphery. Alright, so it is radial and exarch. And the very, very important point. See, how many vascular bundles can you see in the diagram? One xylem and one phloem together is termed as one vascular bundle. So, one xylem, one phloem, one vascular bundle. This is second vascular bundle. This one is third vascular bundle. And this one is fourth. So, there are four vascular bundles, right? So, do remember this very important information. See, in case of dicot roots, the number of vascular bundles will be always 2 to 6, never more than that. Usually it will be 2 to 4, but then because in certain of cases we can see 5 or 6 vascular bundles as well. That's why we have to keep the range 2 to 6. Okay, but usually it is always 2 to 4. So 2 to 6 is also termed as diarch to hexarch. Di means 2 and hexa means 6. So in case of dicot roots, number of vascular bundles will be always diarch to hexarch. It's a very, very important point and a very important distinction between the dicot root anatomy and monocot root anatomy. Okay. Finally, in between the xylem and phloem, in the root, you can see some empty spaces, isn't it? These are going to be filled up with parenchyma cells, which will be together termed as conjunctive tissue. So, in between the xylem and phloem, there remains parenchyma cells, which are called conjunctive tissue. And at last, we have reached the core or the centralmost region, that is the pith. Pith in case of dicot roots will be very small and underdeveloped and inconspicuous and mostly will consist of parenchyma cells. Is that clear? Understood that? Fine. So let me give you a revision so that it becomes very clear to you. So outermost layer in the dicot root is epidermis, and epidermis is going to be made up of parenchyma cells and it is compactly arranged and consists of various root hair. Okay. That's the epidermis. Next, internal to that, hypodermis is absent. So, we have general cortex. General cortex is going to be many layered and it's going to be mostly consisting of parenchyma cells. Parenchyma cells here will allow water to pass through them readily. Internal to the general cortex remains the endodermis, which is single layered, distinct, barrel-shaped cells are present and will possess Casparian strips, which is very well developed. This Casparian strip will not allow water to pass through the endodermis. Okay. Next, internal to the endodermis is pericycle. Pericycle is single to many layered, made up of parenchyma cells, and they will later on give rise to cambium. In case of dicots, it will give rise to cambium during secondary growth phase, which will allow secondary growth. Okay, and also pericycle is responsible for giving rise to lateral root branches. Internal to the pericycle remains the vascular bundles. Vascular bundles are radial, exarch, and always 2 to 6 in number. That is diarch to hexarch. Very, very important point. Next, in between the xylem and phloem, there would be certain parenchyma cells present, together termed as the conjunctive tissue. And the last part is that at the core remains the pith, which is small and inconspicuous and underdeveloped and mostly consists of parenchyma cells. That's it. That is the entire anatomy of dicot roots. Alright, I hope it is very, very clear to you, right? So now let us read it out once so that it is again revised, right? So epidermis, single layered with root hairs. Hypodermis, absent. General cortex, multi-layered parenchymatous, loosely packed, allows water conduction. Endodermis, single-layered, barrel-shaped cells, Casparian strip, well developed, and water impermeable. Water is not going to be passed. Pericycle, one to many layered, thick-walled parenchymatous, helps in secondary growth. Next, vascular bundles, radial, exarch, diarch to hexarch, and pith, small underdeveloped parenchymatous. Right? Very clear? Now let us move on to the anatomy of monocot roots. Alright? Now, in monocot roots anatomy, you will find that 90% it will be similar with the dicot roots anatomy with minute differences. So those differences are going to be very important for you to remember. Okay, so let's start. So as usual, the monocot roots anatomy also starts with a single layered epidermis, which consists of numerous root hairs. Okay, and cuticle or stomata will be absent. Fine. Next, after the epidermis, as usual, hypodermis is absent. So there would be general cortex, which will be multi-layered and parenchymatous and will allow water conduction. Next, endodermis. The endodermis is again the same. It is going to be single layered, very, very distinctly formed. It will have barrel-shaped cells with Casparian strip. Okay. So that Casparian strip will make the endodermis water impermeable. Alright. Next, internal to the endodermis would remain the pericycle. Pericycle, here you find the first difference. All this while you were seeing the same thing, right? There was no difference. Pericycle will show you the difference. The pericycle as usual will be single to many layered and made up of parenchyma cells. But the difference is that here in monocot roots, the pericycle won't give rise to cambium. Cambium is absent. Why? Because the monocots, they do not show any secondary growth. Secondary growth is absent in monocots, which I have repeatedly told you, right? And cambium is going to allow secondary growth. So here, pericycle won't be giving rise to cambium. Alright? Next, other than that, another point that I have told you that pericycle will give rise to lateral branches, that is going to be applicable. Here too, the pericycle will be the part from where lateral root branches will arise. Next, after the pericycle comes the vascular bundles. Now see, how many vascular bundles you can see in the diagram? One xylem, one phloem together makes up one vascular bundle. So how many bundles are here? 1, 2, 3, 4, 5, 6, 7, 8. 8 vascular bundles, right? So here is the major difference. The vascular bundles are as usual radial and exarch, but in monocots it would always be 6 to more than 6, and we term it as hexarch to polyarch. Poly means many. 6 to more than 6. So hexarch to polyarch. That's a very, very, very important difference between dicot and monocot roots anatomy. Alright. Now, apart from that, in between the vascular bundles, there would be conjunctive tissue made up of parenchyma cell as usual present. And the last is pith. Now, pith in case of monocot roots would be very well developed and it will be large. Okay, that's another important difference. Pith here will be large and very well developed, unlike the dicot's anatomy. Okay, dicot roots anatomy. All right, so this is all about the anatomy of dicot and monocot root. So I think you have already understood that the dicot and monocot roots anatomy is almost the same. So all you need to here mark are the differences. What are the differences? Can you recall? First, pericycle. In case of dicot roots, pericycle will give rise to cambium, but in case of the monocot roots, pericycle will not give rise to cambium. Okay, so secondary growth is present here, secondary growth is absent here. Okay, another difference is in case of the vascular bundles. Here in dicots, it is going to be 2 to 6, diarch to hexarch. In monocots, 6 to more than 6. So it will be hexarch to polyarch, right? And finally, the last difference is in the pit. In case of dicot roots, the pit will be small and underdeveloped, but in monocots, it will be large and well developed. Okay, so I hope dicot roots and monocot roots anatomy is very clear to you. So that's it for today's class. In next class, we'll be discussing the anatomy of stem, dicot stem and monocot stem. Okay, thank you.